My training was first at Harvard in the labs of Dudley Hirschbach and Martin Karplus, where I worked on molecular dynamic simulations of protein folding force fields. I went to MIT for the first half of my PhD, working with Angela Belcher on carbon sequestration using yeast display technologies. We're moving to finish up at the Danish Technical University, where I worked on probabilistic models for RNA modeling. There continued at Niels Bohr Institute at University of Copenhagen for my first postdoc with Greston Lindorf Larsen. Then back to Harvard, a postdoc with Adam Cohen, working on optogenetic sensors for live cell imaging, again, protein design sort of challenges, before eventually joining George Church and a friend of mine from my PhD in Angie's lab, Julie Norville, to found JURA Bio, where this was 2017. We're a cell therapy company that makes cell therapies for autoimmune disease and oncology targets. We have a little bit of a novel or innovative mechanism of action that I'll teach you a little bit about today, but hopefully the take-home message is just the way in which a combination of machine learning and synthetic biology makes this, I think, one of the most exciting times in all of history to be a biologist, or to be someone who's interested in taking biology and turning it into therapeutics like I do. So that's my a little bit more earnest background. And so if at any point you want to hear a little bit less about the science and a little bit more about the process of starting your own company, I'm also happy to speak to that. But for now, let me tell you a little bit about this. Generative machine learning, you'll all now know thanks to ChatGPT and thanks to DALL-E. Those are large language models. I have a different perspective on how machine learning and biological sequences interact. And so if for the purposes of this talk, machine learning doesn't usually mean large language model, though they certainly have their time and place. And I just want to give a shout out to my extraordinary team. They're mostly based in Boston in person in a wet lab, but there are also a series of machine learning researchers, both academic and at JURA, that sort of span the globe. And I think that's one of the most exciting things about academia today, but also about businesses today, is that thanks, COVID gave us nothing good except the realization that our networks could be a lot more global, even in a business setting. And so I'm just grateful to the people that contribute to this work. I'll specifically name Eli Weinstein, who's at Columbia, and Andre Nguyen, who's my head of machine learning. So this is the very beginning, and this is the core idea of JURA, my company. So you guys, here's where I admit that my last biology class was literally in sophomore year of high school. And please don't blame me. It is because I went to a girls' high school, which believed that this is a long time ago. They believed that, history and social studies were more important than sciences. That has now changed, thank goodness, but it was a hard thing to do. And when I got to Harvard, I was studying math and physics, so I didn't take any biology there either. But here's my layperson. It's an introduction to therapies for the adaptive immune system. I think a lot of people think about therapies on the B cell side, and you guys, more than sort of anyone, will be familiar with the split and the ways in which this sort of elides over lots of complexities. But basically, a lot of the modern medicines that we make are on the basis of antibodies, soluble or expressed and sequenced from BCRs and their epitopes. We're going to ignore that whole side of the human adaptive immune system when it comes to how JURA thinks about making cell therapies. And that's mostly because there are so many wonderful people making beautiful antibody therapies and moving them onto cells and stuff. Everything I think about at JURA is this other side of the adaptive immune system, and it's the one that's implicated in T cell epitope or antigen display on MHCs. I'll call them HLAs because we're working in the human setting. I'm presuming everyone here is really familiar with the synapse. I just love it. I mean, there's so much of biology that is so beautiful and so mind-boggling, but I especially love the ways in which VDJ recombination leads to T-cell receptor development, and this sort of like ongoing effort of every nucleated cell to chop up its proteins and sort of like offer them up on the surface of the cell. It's just like such an extraordinary process and mind-boggling process. I want to like not lose sight of the wonder that this works and that it works so consistently and so well to keep us safe. And so you will also know this, you know, the T-cell surveillance usually works well. The antigen presenting cell gets infected by a virus or other threat. That little bit of virus is chopped up and presented on the surface of the cell. And all of a sudden, an activated and reactive T cell is able to target and deplete that cell. What happens when it goes wrong, though, and one of the ways in which it can go wrong are in autoimmune diseases, where instead of that T cell recognizing a foreign pathogen or something that should be deleted, like maybe a missense or like a mutation from a cancer neoantigen. Instead, what we have is a bit of self-protein is being displayed, and then that T cell is pathologically pathologically targeting and depleting a cell that actually is yours and that you wanted. Thank you very much. Here is the little bit of this that, like, if any of you are machine learning curious, you know, there is a huge technical challenge, and I think that, like, I really encourage you to get socialized in the challenge of how to collaborate with machine learning peers, or being a machine learner yourself. You know, keep your biology training close to your heart, because there's quite a lot of restatement of simple problems that can be communicated in ways to machine learning audience that make it a little bit more tractable. So in terms of machine learning, or if you're interested in machine learning in the biological space, here's a nice toy mental model. You can think of the immune system as something that's called a classifier. Classifiers are like some of the oldest machine learning models that exist. This is sort of like show a picture of an apple, and this is like, is it an apple? Is it not an apple? Similarly, you can think of the immune system as a classifier that says, you know, the protein's the input, it enters this black box function of presentation and recognition by the T cell receptor. If it's classified as bad, then the attack happens. If it's classified as good, then don't attack it. This is the classifier model of the immune system. Another machine learning phrase is features. So featurization is a process by which you might carve out your data set or data space into objects that are like the components of your machine learning model. And so in this sort of toy example, it's the epitopes that are our features. And so the process of featurization happens on proteins before being sent to this classifier. And so, this is what a machine learning model would look like, that took protein and classified it as bad or good. And if that classifier goes wrong, there are two ways in which it can go wrong. One is that something that was actually bad is classified as good, and that's when you get an infection. And the other time is when something that's actually good is classified as bad, and that's when you get autoimmune disease. And so to bring it back to this idea of therapies, because I fundamentally, as a synthetic biologist, became very interested in therapies very quickly. There's so much that one can do if you stick close to the biology, but sort of move it into unexpected settings that you can do sort of to help human health. And so that's what really gets me excited. So when I look at the sort of therapies that are derived from the adaptive immune system, this is my mental model of what I like. You have the targeting side of the synapse, that might be the antibody, that's the TCR. If you stick it onto a B cell, if it's coming from the B cell, well, these are antibodies or IO drugs. You know, if you stick it onto a cell, that becomes a CAR T. And I know I can't do a raise of hands, but are you guys all familiar with chimeric antigen receptor and T cell therapies?
Raise your hand if you are.
Anyone?
I think about a half are.
Okay, so for those of you that aren't familiar, or those that want to hear it again, it's simple, and here's the framework it tells you. So you take the antibody and you stick it on the surface of a T cell, and now you've redirected that T cell against whatever that antibody targets. So like it's an anti-CD20 T cell that now depletes its target. And so this was really transformative, the work of many, many people, but very classically, the first patient dosed was Emily Whitehead by Carl June around 2014 in a B-cell lymphoma. And this works so well for lots and lots of reasons, but one reason is that antibodies can't traffic maybe to all the places that they should. The depletion isn't strong enough. If you stick that same targeting mechanism onto a T cell, then it almost sort of by definition can travel anywhere that a B cell or T cell lymphocyte can travel, right? And so you get these ectopic lymphs that all of a sudden are cleared of disease. And so it's really transformative. Please, if you haven't read about it yet, after this, go up and look up CAR T or cell therapy, because it's really a beautiful and transformative way to think about medicine, especially for someone who's versed in synthetic biology like me, and so who really likes to make small changes to cells or changes to proteins and stick them onto cells in interesting ways. If you think about using the targeting side of the synapse pulled from T cells, they also are making therapies around this. So you can stick a new TCR onto a T cell. So you're not putting an antibody on T cell, no, you're putting a TCR that was non-native onto that T cell, and now you're sending it out after a cancer neoantigen or something. You're also taking that TCR and you're making a soluble version of it. That's something that doesn't exist in nature, but that we're creating, following this logic of like antibodies are soluble, can be soluble, but they can also be B cell receptors. What if we made soluble TCRs? Well, it turns out they did that. They had beautiful results. I want to say it was in a uveal melanoma, but I forget. It was something so awful, so, a disease stage so late that the patients had no hope, and then here it is, like it's working great. So soluble TCRs. That's also, if we're back in the machine learning world, that's called adding a feature detector, because you're basically adding it's like you're adding to the thymus, right? You're adding something that's going to deplete something. But now let's say we use the targeted side of the synapse. And so now we're talking about the epitope or the epitope HLA complex. On the B cell side of things, this is like you want to deplete autoreactive cells. And so you're going to take linear epitopes, again, stick them onto a T cell, but now deplete sort of the cells that are responsible for pemphigus or other conserved and linear epitope reactive autoimmune diseases. And then finally, and this is why we had this whole long story, JURA is in this funny quadrant that no one else is. We're actually taking epitopes, fusing them to HLAs, and sticking that whole thing onto a T cell. And you're like, Liz, why would we do that? Why could we possibly want it? Well, it's in order to deplete autoreactive T cells. When you get an autoimmune disease, let's say it's MS, and let's say for a second we really believe that all the pathogenic T cells were myelin basic reactive T cells. Those T cells otherwise look very healthy. Like autoreactive T cells in autoimmune disease are usually indistinguishable from other T cells, except sometimes some exhaustion markers, but not reliably. Maybe just based on population, like percentages, but not reliably. Like they're just working T cells that are unfortunately targeting the wrong thing. So when you're in a setting like this, what is the only way in which you can select those cells? Well, it has to be based on phenotype. which means you basically have to dangle a little bit of bait out in front of them. And then if they react to it, like myelin basic protein, you dangle it out to them and they react, then almost by definition, that's a pathogenic cell, right? Because the amazing thing about immune surveillance, TCR immune surveillance in your body, is that if you dangle any piece of your own self protein, the TCR should ignore it. You know, like it doesn't matter where in the body it comes from. And so that's what's extraordinary. And that's what's so exciting about making these HLA CARs. Now, that introduces a whole big problem. It could be easy, could be hard. In general, you'd generically like to be able to move from, observed sort of T cell population into what epitope HLA it's reactive to, or vice versa. And guess what? This problem isn't solved. And I will now walk you through. Oh, gosh, I have 12 minutes left.
Oh, you have to 12:45.
Oh, great. Oh, great. Then I will now walk you through how I think about complex problems like this. And again, I'm encouraging you to ask me questions or stop me at any time, or if it's getting boring, stop it. But here, let me tell you our challenge problem. So the T cell recognition problem is an open problem. It is a lower affinity complex than antibodies and their targets. It is something that is at such combinatorial scale that I love. People always make these, these comparisons to like numbers of grains of sand and stars in the universe and blah, blah, blah. I can never keep them straight. It's just like so beautifully complex that I know that like if we were to sequence our own sort of individual person's immunome, sort of TCR diversity and BCR diversity, that's like 10 to the 4th more data than in our genome. And so it's just like a beautiful challenge. And that's like each person has their own thing of this. And so like I always like thinking about orders of magnitude. So like you maybe have 10 to the 11th T cells per person. You guys probably had to memorize all this stuff for USA Bio. I don't know, but I always love having sort of like order of magnitude approximations whenever I confront problems. But you maybe have, of those, 10 to the 8th that are unique clones. So you could expect, if they were randomly, evenly distributed, 10 to the 3rd, about 1,000 of each TCR clone. Now that isn't the case. What you actually find is you have super rare T cell receptors and you have super common ones. But, this is the number of clones that you have out of, gosh, the estimates are huge, but like 10 to the 15th, 10 to the 17th, they're possible. On the other side of this challenge, you have the epitopes and the human HLAs. Now, HLAs are this combinatorial problem, inherited problem in their own sense. The epitopes themselves are derived and processed. If you look at the peptidome, you're talking about, I think hundreds of millions of processed epitopes. If you're looking at UniProt, or the entire sort of, not just human, like proteins from which to derive HLAs, we're looking in the trillions. It's really beautiful, hugely complex. And we're planning to solve it through a combination of machine learning and synthetic biology. And so here here's just like if you were talking to a machine learning friend, how I would explain it. A function that takes a set of protein sequence spaces, each one being the TCR, the epitope, MHC, maps it to a complex and gives a probability of that complex or strength of binding. But, I will take a step back and say, how would solving this problem aid medicine? And I know that some of the therapies that I talked to you today about were maybe new to some of you, but I will encourage you to think, about this for other problems too. Like always think like, gosh, here's a beautiful biological problem, try to keep aware also of how it might be employed in aiding human health, because I think that the gap between biological discovery and translation is becoming narrower and narrower in time. And so I think it's both interesting and rewarding and compelling to keep that in mind. I am I really believe deeply in basic research as well, and for its own sake and for the beauty of it. But I also just say, gosh, if you're someone who really likes translational medicine, this is a wonderful time to be doing both basic research and translational medicine at the same time. The gap is closing. So we've now solved this problem. How would it help in medicine? Well, we could help with diagnostics. You could get the TCRs that were effective against certain antigens. You could get the TCRs that were ineffective, so you could like diagnose autoimmune diseases better. In general, and I love this point, you would obtain a long-lasting record of all past disease exposure, everything that a person has been through. The adaptive immune system sort of keeps a record of in this beautiful way. And so if you were able to decode that, you basically get this beautiful record. On the therapeutic side, and here's like an alphabet soup of all the new therapies that are coming out, and I'm always happy to answer any questions about any of these, but adoptive cell therapy, TCRT I mentioned, TREGs, it's like TCRT, only now instead of a cytotoxic T cell, it's a regulatory T cell, so you're inducing tolerance. TCR bispecifics, this whole other thing. Vaccines, Moderna has these beautiful N-of-1 cancer vaccines that are doing wonderfully in clinic, and there's lots more programs where that came from. And then finally, my own weird part of the university's EMHC CARs for detecting and depleting pathogenic T cells. What's the challenge? You have a data challenge. And this is true whenever you're thinking, and I will encourage you to think about this, because I know that you as scientists are going to be doing more and more machine learning, even in your day-to-day. Even if you want to be the purest of biologists, I encourage you to think about things from this perspective. Because there's often a data challenge, and in biology, it's almost always a data challenge. We think of these as large data sets, but actually compared to like all the likes on Facebook or all the Instagram like hearts, we just don't have that much data in biology. It's expensive and hard to get. High throughput sequencing gets us into a regime that's like this, awkward middle between like too big to do easily, but too small to actually just work really perfectly with a lot of the machine learning models that have developed for serving ads. There's the model challenge, and this is the type of machine learning model that you bring to bear. And I will say that usually for biology, you want models that account for experimental biases and that match something that's called the out of distribution challenge, which is in biology, we only usually see a small fraction of the sequences or the problems that we're interested in. And so making sure that you have a model that generalizes beyond just observed data is really, really, really important. And so I'll talk about that. And then finally, this outstanding challenge, and this is one that's particular to me, not of solving necessarily the biological question, the basic biology question, but like actually I'm planning designing therapies. So sometimes the machine learning models that work for solving basic biology problems are not optimized for solving a question of, okay, what's the best therapy that I can give this person? How can I design it and make it best? And so always be cognizant that you are solving the right sort of challenge. It's easy to get caught up in like the wrong choice of model because you forget that you're trying for design or you're trying for basic biology.
Okay, have a question?
Great, please.
Bill?
For machine learning models, I know a lot of nowadays, like other kinds of models use like multi-layer perceptron or it's like mirroring neurons. But for this specific kind of challenge, since it's like accounting for biological proteins and stuff like that, is there any specific like cases where you would want to use biological models like feedback models to change your machine learning model?
So lots of so I'll answer your question two ways, because I am very sensitive to the fact that a lot of the machine learning models that exist, like neural networks and things, are in some sense, or perceptron models are like biologically derived. But I don't think that's what you mean by biological models, right? You mean more like a feedback loop between testing and lab, right? So that to me is my ideal case. Like I like am in the world of having tight iterative loops. It's a world called Bayesian optimization, where you're doing a handful of experiments. You're making a handful of sequences, you're testing them, you're feeding that back into the loop. It is suggesting the next 10 sequences to do, and then this comes back down into the design, whether you're looking what your model challenge is and what your design challenge is, because like in a lot of these iterative design loops, you could either be looking to explore the space completely, that's usually a more biological challenge, or it could be just trying to get to one thing that works pretty well really quickly. And that's often more where you are in the therapeutic side. But I absolutely think that this integration of data and feedback into your model is sort of crucial for intelligently moving through your search space. Otherwise, you're stuck in these grid searches, and it's just like so wasteful and costly. You're producing so many sort of like sequences that you're then testing, and you're not getting as much information out as you should be. So that was an excellent question. Thank you. Let me know if I should answer any more.
Other questions?
Yeah.
Okay, Michelle?
All right, so I wanted to, so you're talking about how the machine learning was used to optimize and make certain or new models, for example, like CAR-T, et cetera. So I wanted to ask, how specifically have you used machine learning to optimize CAR-T, or how, like what instances has machine learning been very like useful in optimizing it?
Yeah, so I'll give 3 worked examples. I mean, we can get there. This is I'll show you exactly how we're doing it, but I'll give you 3 examples. For other people, building sequences at large libraries, exact sequences at large libraries, they might want to make and test 10 to the 7th sequences, and that might cost them $100,000 to $300,000. We developed a process called variational synthesis, which is a machine learning optimization of synthesis procedures. So we can make 10 to the 14th sequences manufactured for $1,000. That's a 10 to the 9th reduction in cost, which means that for us, making exact sequences and testing them is no longer the limiting factor in any of the work that we do. It comes down to sequencing costs on hits. Another example is work with Katie Rizwani. She's at MD Anderson working hard on CAR and K cells. So this is, rather than using the T cell as your effector cell, you're putting a natural killer cell in. That's nice because they don't expand as much. And so you might not get this cytokine storm that can sometimes go wrong in CAR T dosing and administration. So she had the question of, gosh, we're testing many parameters in our pathway between in vitro tests, mouse samples, and then finally like patients and patient outcomes. She had 2 questions. Which parameters are actually the most predictive, and what regimes of those parameters are the most predictive of success? So that's actually 2 separate questions, right? Which parameters contribute the most to one outcome or the other? That's like a basis set question. But then the other question is like, what's the optimal ranges for those important parameters? And so we were able to set up models for her, and I mentioned her because we're not yet into the clinic, but this was clinical patient data, that predicted the lifespan increases based on sort of those model parameters. So we're able to see, okay, these are the measurements you should really make, and then these are the optimal like values for those measurements if you're going to take something into clinic. And so that's number 2 for machine learning. And the third example I'm going to talk about in this talk. So you'll see that in a second.
Thank you.
No problem. Okay, I'm going to talk a little bit more about model challenges because I really like this. You need to account for sources of bias. Many public data sets lack negative examples. So like, just keep this in mind. There is a whole field of machine learning on how to learn from only positive examples. Like, imagine if you're knocking at doors and polling people, and the only people that you get answers from are the ones that answer the door. Now imagine if when you went to publish it, you didn't mention how many like doors you knocked on that no one answered, and you only sort of reported 100% of households, say this or that. That's how biology often works, right? Like we don't count or sequence things that aren't hits, or so we're really in a regime where we almost always have negative examples. We also have lots of noise. Lots of people talk about this, and there are more or less sophisticated methods for dealing with like one technician does this thing, one technician does that thing. One day it was humid, one day it wasn't, that sort of stuff. Handling biological sequences. Almost all the existing machine learning models that you have access to have been optimized for language, for images, for networks. None of those share the rare and beautiful statistical properties of biological sequences. And so the sorts of biological sequence machine learning models that I put together are a whole graduate education away from where you are and where you will be. But like, go study some machine learning, and when you're interested in this, come back to me, because I'm very happy to sort of set you, if you're interested in this, set you maybe on what I perceive as the right path for building models that are specific to biological sequence. And then finally, this distribution shift thing that I talked about, which is that often the sequences we care about are far from the sequences that we trained on. And there are ways in which you can just shoot yourself in the foot, because the model works extremely well on everything you've seen, but you don't care about stuff you've seen. That's stuff that's already been measured. You want to know about this new TCR that you saw in a new patient, and you want to know what its antigen specificity. And so if I look, if I think about the next thing, it's this mindset shift from just biological sequence prediction around the TCR HLA problem, the design of therapeutics. And so I think this will, for the people that are interested in translation, let me do this quickly. Prediction isn't enough, right? You need an optimized strategy. You're not going to be able to do everything, and so you want to search through a high-dimensional space quickly. You have a different sense of uncertainty. You'll want to avoid creating and testing sequences with very high uncertainty, even if the expected binding is good, because it's just wasteful of your resources. So you'll want a higher certainty, sort of better binder when you get the choice. Variety, you want it to reflect the diversity of the human population and not just the biased biological sequence data that we often have. The human, until recently, in the Pangenome project, our reference genome was a white dude in Cleveland, which is where I'm from, which is why I can say it with disdain. This is not reflective of the beautiful genetic diversity of humans. And so like the idea that we're building therapies around this one person's genome is just terrible, but that's coming along. From the therapeutic design challenge, you want to be able to manufacture lots of challenges. That's what I was telling you about with variational synthesis, which brought down our costs by 10 to the 9 orders of magnitude. And then you also want to be careful about correlation versus causation. And so like there's a whole field sort of started by Yuta Pearl, but of causal machine learning, causal inference. And so for those of you who are interested in that, I'll just like say that keyword and go search causal inference. Okay, finally, the data challenge. And you asked me what's like the most important machine learning challenge that we're going to solve as a company, or maybe even as individuals. It's overcoming the data limitation challenge of the epitope HLA TCR prediction problem. So there have been lots of decades of beautiful work going from a single known epitope in HLA and then getting bunches of TCRs that are reactive to it and sequencing the TCRs. There have been lots of things in the other direction. You have a known TCR, and now you're going to do yeast display and do a lot of epitope MHCs and get all those sequences. What has eluded us and continues to elude us is the ability to get bijective information or sequencing results on both sides of the synapse. That has highly biased and kept us from moving forward with our ability to work on this challenge. So people have been spending a huge amount of time coming up with more and more complex and beautiful models. So we go back to that. We go back to this framework. They've been focusing on the model part of it and trying to extract as much information as they could from, okay, just a one epitope with lots of TCRs and one TCR with lots of epitopes. But the truth is that they've been stymied because this introduces all the problems that I talked to you about. There's this out of distribution problem. There's these bias problems. There's, all of a sudden you get an unknown TCR and 9 out of 10 times they're going to say, oh, it's reactive to influenza, CMV, or EBV. And that's not because that's what it's actually reactive to. It's just because that's the only data set that we have. I think we have representation of 1/10⁸ of the possible, sequencing results on one side of the synapse. So if you combine that, we have 1/10⁻16 or more. It's just like we are in the data limited challenge. And so this is why I bring up these rubrics to you, because never, when you're attacking the new problem, never lose sight of what is actually your overriding problem. Lots of the beautiful scientists that have done beautiful work on developing better machine learning models have sort of ignored the fact that, listen, yours is not a model problem, it is a data problem. And so if you're actually going to set out to solve this problem, figure out how to make machine learning help you on the data side instead of a more robust model for learning from small data sets. So this is going to come, so variational synthesis was our answer to that. And so I will skip it, but I wanted to give you sort of a sense. For me, as I was starting my journey into math and physics, it was very helpful to be sort of introduced to the language and the look of work that I wasn't even familiar with or ready to process. And so I just want to sort of like show you how a machine learning talk looks. We compare it to stochastic synthesis, which is your other way of doing very large libraries. Our new method, we write down a generative model of the actual synthesis protocol. So I'm actually working with manufacturers and actually creating a generative model around like physical parameters. I'm minimizing divergence. That's something you do in almost every machine learning problem. And then I'm running the synthesis protocol in the lab. So I'm like going from lab to computer to machine learning to output and then resynthesizing it again. And so this just allows me to make these extremely large libraries at extremely low cost. And it is only because I was able to correctly think about what my problem really was, that it wasn't about coming up with an even more refined or successful machine learning model. I could make it as sophisticated as I wanted, and I still couldn't overcome the challenges that I had set for myself. So that helped me very quickly get to the point where, okay, this is actually a data problem, and I'm going to have to use machine learning to create better data. And that, I think, is what a lot of people lose track of. You can actually use machine learning to get better data. You don't just have to keep it in that box of the model itself. Like, be innovative and attentive to how you think about data challenges, and make sure that the data you're collecting meet your design, goals. They meet the question you're trying to answer. Don't sort of get into this path of doing something just for the sake of doing it. Always make sure that you're doing that, you know the part you need to push on the hardest, and then you're pushing on it with all your might. Okay, that's it from me. I am so happy to take any questions and also like tell you anything.
Any questions? Okay, Bill.
For someone looking to like do sort of this school biology ML sort of a career, do you have any suggestions for what someone should focus on learning?
Gosh, you know, it's a really good question. And I have to say that I am very persnickety and nitpicky about who I think is doing this well and who is not doing it well. And it is so hard to be sort of an autodidact in this area. But I will point you to, there's a guy, I'm going to put it in the chat, but maybe you can look him up after. I think Fabian Tice is doing really beautiful work on creating a whole online open source textbook of the absolute best sort of like fundamental machine learning models and thoughts about like careful handling of biologically relevant machine learning. And so I'll send to Kathy the link to that textbook after. I think that is like great preparation and will inoculate you from a lot of people who are saying, oh, we solved it. We did the LLM, you know, we solved it. You know, they don't know what they're talking about.
We just listened to another very, very happy scientist. Okay, so it's so good to see that enthusiasm and joy just come right out of you with what you do.
I have to admit, it's been 7 years that I've been building JURA, and it hasn't always been this good. You know, there was a desert period where it felt like everything was hard, and we were building this entire tech stack, and no one even saw the need for why we were doing this tech stack, because they were saying, oh, it's a data problem, just go out and sequence a bunch of stuff, like just build a lot of stuff. And I was like, you know, oh no, we can't possibly build. This is that order of magnitude thing I said. You know, I set out and said, gosh, I can't get to the order of magnitude I need with current methods. So that's what set our whole research path, and that's what made it possible in the end. But there, I don't want to give the sense that this was easy. This was, you know, there were definite moments of self-doubt.
Yeah, but positive as well. Thank you so much, Liz. We really
my gosh,
appreciate it.
It was so wonderful talking to all of you, and thank you. Next time I hope I get to see you, but thank you so much.